Hey, welcome to Unknowns. So today we are going to see about endophytes. It's been a lot of talking about endophytes, especially during summer internship this time in phytomicrobiology around 2025. So what are we going to see is why so much significance associated with endophytes? Why to study endophytes? Okay, let's see. So endophytes actually, they are the means of plant survival, I would say. They help plants in many, many things. Example, in phytohormone production, in cererophore productions, in antibiosis. Endophytes are the one actually, control pathogenic microorganisms that can infect plants, otherwise called as phytopathogens. They increases or strengthen the plant immune system, and they induce resistant against various infection and drought resistant, stress resistant. They play a fantastic role. So basically, what are endophytes? The microorganisms that are living inside the plant tissue. Okay, maybe between the plant cells or inside the plant cells. When I say endophyte, I meant endophytic molds, yeast, bacteria, actinobacteria, both eukaryotic and prokaryotic microorganisms. Okay, so why is so much importance is associated with endophytes? Here is a proof. Diversity of antimicrobial secondary metabolites produced by various fungal endophytes. Okay, they are producing metabolic byproducts that can inhibit other bacteria and fungi, so they can be effectively used in disease control. There is another endophytic utilization. See, fungal endophytes, a modifier of plant diseases. It is well proven fact that a plant with diversified endophytic population inside their biological system actually are more immune to various diseases. And here is another paper that compiled the natural antioxidant produced by various endophytes. And the highly isoprenoid lipids, oxygenated isoprenoid lipids, they are also highly antioxidant in nature. You should know any compound that is more antioxidant in nature is also anti-diabetic, anti-cancerous. They are associated with many other good functions. Okay, and here is an interesting paper where an author is advocating that we can use endophytes to increase the shelf life of fruits. Post-harvest disease, spoilage because of various microorganisms is a common scenario that leads to a lot of economic loss. Here, the author beautifully described how the fruit life shelf life can be increased by coating, injection. Many processes are there where these endophytes are known by this plant system, and the endophytes know the plant system very well. They're not going to spoil it. In fact, they are going to increase the shelf life of the fruit, and they prevent the fruit and vegetables from spoilage. It's actually a very interesting paper. And here is another paper. This is much more interesting because they have developed, or they tried to see how to make an endophyte as entomopathogen. Because when I say entomopathogen, any microorganism that can infect plant pest. Okay, they can control the insect population so that agriculture can be benefited as an entomopathogen. When you develop an endophyte as an entomopathogen, imagine, the common problem with the entomopathogenic fungi is they are less resistant to UV, and with they need more moisture. If there is a less moisture in the soil content, they may prone to they may decrease their effectiveness against insects. But if they can occur as an endophytes, they can, when they are infested upon by insects, they can act as entomopathogen also. So the lot of endophytes also have nitrogen fixing capability. They have been used as an biofertilizer. Even entomopathogens are used as a biofertilizers. So this paper tried to see if we can use a endophyte as entomopathogen. Okay, so why? Because most of the endophytes are known to be a biocontrol agent against phytopathogenic fungus and other phytopathogens. Okay, because they also can produce a lot of antifungal compounds which can produce other fungi. So that's why it's very important to study endophytes. So any fungi after surface sterilization of explant, when the fungi comes out of the leaf, usually the endophyte will come out of a explant. This is a plant leaf that has been inoculated perpendicularly to the surface of the agar. The endophytes are coming out. Okay. So commonly, the endophytes are isolated like this. We have both uh, culturable and unculturable methods. Okay, you can either the segment can be sterilized, surface sterilized and inoculated in the media, or the segment can be emacerated, that can be mixed, and the water after surface sterilization will act as a negative control, and the emaceration can be plated. Okay, a lot of ways we can do. Otherwise, if you want to assess the entire endophytic population, simply extract the total genomic DNA or total DNA from the leaf tissues. From that DNA, you can screen for bacteria, fungi, whatnot. Many isolations can be done. So basically, plant leaves are collected, surface sterilized, and the sterile leaves are actually imprinted to see the effectiveness of sterilization, and they are inoculated. Once the molds are coming out or any fungi or bacteria are coming out, they will be pure culture. They will be biochemically, morphologically, biochemically, and molecularly characterized to know what species it is. Okay. So this is a basic flow chart, collection, external sterilization, and endophytic culture. Axenic culture is a pure culturing. Okay. Culture plates containing single species of endophytic fungi. So this is the flow chart of common molecular methods where DNA is extracted, PCR sequencing. Using bioinformatics analysis, we will find out what species it is. Okay. So you will see when you progress with endophytic isolation, some can be done with axonic culture. Some endophytes demand co-culturing because one of the main vital products for their colony formation depends on the metabolic byproduct of its neighbor. Okay, the neighbor has to give that metabolic byproduct to form a colony. We will see that in course of our internship. See, endophytic population is highly dynamic. Okay, it is plant taxonomic specific. Even plants have two groups of endophytes: core endophytes and accessory endophytes. Core endophytes are endophytes that will present only on that particular plant species, and accessory endophytes are endophytes actually will occur commonly in all plants occurring in that particular geography. Okay, so yeah, it is geographic specific. Even the same plant species, if you sample endophytes in summer, it will be different. The diversity will be different, and in monsoon, the diversity will be different. Yes, weather matters, climate matters. Okay, and season matters. Same plant species occurring in high latitude, low latitude, high altitudes, you will see the endophytic population differs. Okay, and even when are you collecting? Say, for example, in the agricultural land fields, immediately after watering and after 2 days of watering, if you collect the leaf and check for endophytes, you may end up isolating different diversity. Okay, so that also depend on inter and intra-species variation, how the plants are, or what kind of plant your target plant is having a relationship with. But like what kind of plants are growing around your target plants can also influence the endophytic population in the target plants. Plant age. When you sample a particular plant leaf in when they are 2 years of age and 20 years of age, definitely they will have different endophytic populations. And type of plant tissue. Are you going to isolate endophytes from the fruits, leaf, stem, flowers? Yeah, endophytic population differ despite it's been same plant species. Okay. So not only that, in which side you are collecting plants, that plant leaf. Okay, say for example, when you are isolating endophytes from the plant leaf, uh, see, when you sample a leaf from apical meristem or lateral meristem, the endophytic population differs. Okay, and even right side sampling, left side sampling, which side is a east side or west side, endophytic population can differ. Even the fragment dimension. Okay, when you do the fragmentation with the punching, paper punching, you can do it and have an endophytic disc. Different disc size, uh, they may influence the endophytic population. And from where are you collecting in a transect? If you want to assess a transect, this is the way to plan your sampling. Like I said, what kind of neighboring plants are there can also influence the endophytic diversity in the target plant. So uh, the endophytes are of very different types, and also what kind of surface sterilant you are using. Basically, in all endophytic isolation, they will be using a ethanol as a basic surface sterilant. And there is a main sterilizing agent where sodium hypochlorite or mercury chloride can be used. Finally, whatever sterilant you are using, that will be treated in distilled water, washed off, and they will be taken. Even though many protocols says 95 ethanol for 5 seconds and sodium hypochlorite for 2 minutes, I think 2 minutes is too much time. Maybe because of 0.5% hypochlorite. You can have 0.5% for, I would say, 10 seconds, not more than that. And another ethanol treatment in different concentration. Even the order of surface sterilant that is used for sterilizing the explant will also influence the endophytic diversity. Okay. So what I'm trying to say is it is very hard to have a constant diversity while working with endophytes. Okay. So either you wash the leaf in final distilled water and use the distilled water as a negative control, or you imprint the leaf, or you do the both to make sure that the surface sterilization works. So these are the common surface sterilant usually used for endophytic isolation: sodium hypochlorite, mercury chloride, hydrogen peroxide, 1% peroxyacetic acid, calcium hypochlorite, sodium hydrogen carbonate, and sodium azide. Okay, even 2080 are also used these days. So, is it necessary to use the chemical surface sterilant? Not always. We can also use sonication, UV radiation to physically knock out the epiphytes, that are the organisms that are living on the surface of the plant tissue. Even you can conduct a mock experiment to see, spray a E. coli in the leaf, expose it to UV, different timing to check which timing is better for eliminating every bacteria on the surface of the leaf. How to know that? After treatment of the UV imprint in the EMB plate, probably eosin methylene agar plate, to see if E. coli has been completely knocked off or not. Such a mock experiments can be conducted. This is what been conducted here. Here, the leaf collected is being imprinted on the surface of the agar plate. Okay, and here the leaf has been sonicated and done here ethanol sterilization, sodium hypochlorite, that is bleach sterilization peroxide. Looks like sonication bleach peroxide is working much more better than ethanol. Uh, the what you can understand from this is different plant species may require different methodologies to efficiently remove the endophytic microbiota. Yes, if you are doing a dedicated research on a particular plant species, you need to optimize your surface sterilization protocol. You need to find out which protocol works better for you. Here is a transmission electron microscopy clearly showing microbial cells, fungal hyphae near the stroma, and other plant parts to indicate that endophytes are actually living inside the plant tissue. Okay. So this philosophy works better for endophytic isolation. We reap what we sow. Okay. What kind of media you are using will also select endophytes. Let's see this very clearly. When you do a surface sterilization, it is not only surface that is being sterilized, even the surface sterilant will penetrate into the plant tissue and they are going to influence your endophytes. Whichever endophytes can withstand that particular chemical for the exposure period, they will actually regenerate and they will come out. Okay. How or what kind of endophytes, can all resistant endophyte come out in the media? That depends on what kind of nutrient you have used in the media. Even though potato dextrose agar is generally recommended, actually malt extract agar is known to harvest more endophytic diversity than potato dextrose agar. Chapados dextrose, we have tryptone, water agar media, simply agar plates without any nutrient. You can use it for isolating or wooing the fungi out of the surface sterilized leaf. And as we all know that molds prefer slightly acidic pH. Okay, if your pH is around 5.8 to 6.5, usually molds can come out very easily. Okay. And some studies have also suggested supplementing the host plant material to culture media enhances endophytic recovery. This is something very interesting. Okay. I remember a study that just collected the plant leaf, heated it in microwave oven, extracted the soup, they have added agar-agar to it when for plating. And they claim that whatever diversity missed by potato dextrose and malt extract agar, actually this innate nutrient that is being incorporated in the media actually facilitated the endophytic isolation. I think we should also try this. Some plant species usually culture more endophytic bacteria than molds. When you are targeting your mold isolation, these bacteria are big hindrance. In that case, you can use a antibiotic cocktails, but broad spectrum of antibiotics to inhibit bacteria, to stop bacteria growing out. Okay. So yes, the cultivation condition. If you incubate your plates in 28 degrees Celsius, mostly facilitate fungi. If you incubate more than 30, 32, it will facilitate endophytic bacteria coming out. Incubation time. I remember a work that incubated the plates in 30 degrees Celsius, which is focusing on bacteria, endophytic bacteria for 3 days. Whichever bacteria came out, they have subcultured and then they transfer the plate to 35 degree for another 3 days. Likewise, they played with the temperature to harvest more bacteria. Light is a key factor for sporulating the endophytic fungi. You will see 50% of endophytes will not sporulate. So what's a big deal about sporulation? If they do not sporulate, their taxonomy becomes even more difficult. Okay, so sometimes light exposure can induce sporulation, and we can ease off our taxonomical effort. So the common size of explant to be inoculated would be 5 millimeter square. Okay, 5 into 5 millimeter. That can be a common plant size that can be used for the explant inoculation. Okay. So once you inoculate it, it is very much the immediate step is succinic culture, that is segregating the endophytes based on their color, texture, topography, pigment. Pigment will be a diffusible pigment, non-diffusible. Everything should be considered for characterizing or selecting the endophytes for pure culturing. Okay. Preservation, endophytes can be preserved by numerous methods. We usually use agar button method described as the last step. Just grow them in the plate, take a plug, put it in distilled water, 4 degree or room temperature can do. But if you are going to preserve it below 0 degree, better use glycerol in them as a cryoprotectant. That's what been discussed here. Okay, minus 20, minus 80, minus 180, better use glycerol stock. And for molecular identification, the common step is because most endophytes do not sporulate. Now, molecular identification becomes very vital for them because the sporulative structures are usually a key characteristics features for many species level identification. Because they do not sporulate, now we have to extract DNA, target ITS region during PCR, and whatever amplicon we are getting, we will sequence it by bioinformatics and blast analysis. We will know what species it is. Why we don't call them as an ITS gene? We call them as an ITS region because they are combination of 5 genes. The last fragment of single small subunit RNA, ITS1 gene, 5.8 gene, S8 gene, ITS2 gene, and the initial fragment of large subunit, that is 28 sRNA subunit. So this is a primer commonly used to segregate out the 650 base pair of ITS region, 650 to 750. It is variable because you should know internal transcript spaces are frequently subjected to insertion and deletions. Okay. So ITS1 and ITS4 is a common primer region that is used, and that will bring out this region as an amplicon. So more than 97% of similarity can be considered as a closest match. That can be considered the same species. The species cutoff is 3% here. Anything over 94 and less than 97 can be a genus cutoff. So I want to quickly discuss about a thesis, PhD thesis, which targeted isolation of endophytes from mangroves. Mangrove forest. Those are coastal plants that can grow in coastal waters, seawater. Okay, so the author has used two different methods, 70% ethanol and 75%, then treated with 4% NaCl. You see there we had 0.5% in sodium hypochlorite, which is for 2 minutes. When the concentration increases, okay, no, yeah, that is for 2 minutes. They have also used 4% for 1.5 minutes and 2 minutes. Okay, and finally, distilled water sterilization or one more ethanol, and they have inoculated in the PDA media. Usually, when you use a coastal plant, it is very important to prepare a PDA media using the seawater. So that media is prepared in seawater, and these endophytes are mostly marine fungi. What is the main difference between marine fungi and terrestrial leaves? Marine fungi require NaCl concentration for their reproduction. Any fungi that tolerate NaCl cannot be considered as a marine fungi. They may be marine tolerant. All marine microorganisms require NaCl for reproduction, okay, for their growth and reproduction. So these are the common fungi: Alternaria, Acrimonium, Cladosporium. They have isolated out of the mangrove leaf. This study is done during 1990s, I guess, but still this picture is very much intriguing for me. You see, this author has prepared a picture. Actually, this is Dr. Kumaresh, and a famous mycologist. He is in associated with the Pondicherry College, Kumaraguru College, I guess, in Pondicherry. He's a famous mycologist. This is his thesis. This author has drawn a picture of a leaf and segmented the picture. You see, starting from 1, 2, 3, 4, 5, 6, 7, 8 is segmented, and he inoculated the respective pieces into the PDA media prepared in seawater because it is mangrove species, and that explant has been named. One is from this region, 2, 3, 4. Okay, so if you watch this leaf structure here carefully, there are single dot. Say for example, in segment 46 and 47, you have a single dot. 53 and 54 have two dots. What these dots means is only one fungi came out of this explant 41. When you have a two dot, two fungi actually came out of 53rd segment. Okay, all those segments can be tracked like this. It's really a very interesting way to see. Actually, the central part, the rib part, is actually marked here in different color, and they have segmented the entire leaf and they have named the explant. Okay. So once you have a fungi isolated, you have to pure culture, agar preservation. How to see if your fungi can control a phytopathogen? How to see if my endophyte can fight against a disease-causing fungi, plant disease-causing fungi? Just put both the agar buttons in the same plate and see the pattern of growth. See here, this endophyte secreting something, not allowing this plant pathogen to come near. The growth is actually arrested here. So like this clear pattern usually indicates the, see, it is very much more clear in the E section, E picture. This endophytic fungi is not allowing this phytopathogen to grow more. So these are a clear indication that your endophytes have an antifungal property. Okay. And phosphate solubilization. You know, phosphate usually occur as an insoluble form. Plant needs help in solubilizing the phosphate and make it readily available as a bioform or absorbable form. Okay, so in phosphate solubilization experiment, these phosphate plates are actually opaque. When an endophyte produces enzyme phosphatase to solubilize the phosphate, or they produces many organic acid, which in turn make the phosphate into soluble forms to be that can be easily uptake by plants. They will produce a clear zone around them. So these are phosphate solubilization experiment to see whether fungi has a phosphate solubilization activity or not. And here we have used UV spectrophotometer to clearly distinguish the endophytic species. We are not species. You can say morphotypes or type species. We are yet to declare the species. Okay. Say for example, this endophyte is producing a pigment in reverse plate, but it is not a diffusible pigment. So media remains the same color, and this produces yeah, diffusible brown color pigment. Okay, so we grow them in the potato dextrose broth. We centrifuge it. Cell-free broth we subjected to UV spectrophotometer from 200 to 2100 nanometer. Four different among the 9 endophyte species. The broths we subjected, I could say we had actually four different species based on the metabolic fingerprinting. The peaks we can observe during the UV signature screening or UV screening, we can clearly say there are four unique peaks corresponding to four different fungi. Now you can subject only these fungal plates for ITS sequencing. Okay, that's how we got all these fungal cultures. It is all available for commercial exploration and for academic research also. You can check this website if you want. You can order the fungi from. Thank you.